Welcome back, and in this video we're going to take a look at one of the key techniques in cell biology, and that's immunofluorescence. We're going to look at a lot of these techniques because when you get onto 4,000 level classes and you are looking at data, it's important to understand how that data was generated and whether the technique that was used was appropriate for generating the data that's being analyzed. So immunofluorescence, there are a couple of different ways you can do this technique. This is a visualization technique that's used in combination usually with confocal microscopy. Remember, that's the microscopy where we have a labeled cell, and here we're going to label the cell using immunofluorescence, and we take thin slices, optical slices, by focusing on one plane in the sample and then slightly adjusting the microscope and taking another image in another different optical plane. Then you can stack those all together and you get a very high quality stacked image. So how do we do this technique? Is what we do is we get some cells from a sample, and we could talk later about how we get those cells from different places and different samples. And we take the cells and we get them onto a slide, a microscope slide like you've seen and used before probably. The cells are then treated with some different chemicals to fix the cells in place so things don't move around, and to permeabilize the membrane so that molecules can go in and come out. The first thing that gets added during immunofluorescence is an antibody. Remember, antibodies are generated by immune systems, and they are specific for certain proteins. Usually proteins, it can be for other things, but we're focusing on antibodies specific for proteins. And so you can take an antibody which recognizes a specific protein. Let's just say it recognizes tubulin. And so this antibody will bind to all of the tubulin in the cells. And like you know, tubulin is used to make microtubules. And so this antibody would be stuck all over the surface of the microtubules. We then wash away all the excess antibody that's not stuck on the microtubules, and then we add a second antibody, and this second antibody is an antibody which recognizes the first antibody. So it's an antibody against an antibody, and so the second antibody goes into the cells, into these fixed and permeabilized cells, and it binds to the first antibody. So we've got kind of the microtubule with an antibody stuck on it, and then another antibody stuck on the first antibody. The second antibody that gets added, the one that's binding to the first antibody, that has a fluorescent molecule on it. So this is called immunofluorescence because we're using antibodies which are derived from immune systems and a fluorescent molecule. So what we end up with is what's called the primary antibody here attached to our target protein. Perhaps it's a nuclear envelope molecule or something like that. The antibody that we first add is the primary antibody, and it binds all over the target, the thing to which the antibody binds. Then we add in a second antibody which recognizes the first antibody, and this second antibody is tagged with a fluorescent molecule. And so when we excite this fluorescent molecule on these antibodies with a laser, wavelengths of light will hit the fluorophore, and then fluorescence of a different wavelength will be released, and we can use some fancy filters in the microscopes to eliminate all of the light except the wavelength we're looking at. So what we're seeing here is a dual labeling. So this is two fluorophores being used here. The first thing that's been labeled are the microtubules, and they've been labeled using immunofluorescence, and so the first antibody recognizes the tubulin, the second antibody with the fluorophore binds to the first antibody, and when we illuminate that with one wavelength of light, it glows green, and we take a picture of that. And then what we've also done here is we've added a dye which is able to bind to DNA, and so we see the nucleus and the nucleoli in orange here. All the little tiny dots are probably the mitochondria or just background. So we take a picture of that, and then we put the two pictures on top of one another, and we have this compound image of all the microtubules throughout the cell as well as the DNA. Let's take another look here. A slight variation on this, which is not immunofluorescence, but as a variation of fluorescence microscopy. You can also tag or label proteins of interest with fluorescent proteins. You've probably heard about the green fluorescent protein from jellyfish. And so you can isolate the DNA for that jellyfish fluorescent protein. You can stick it onto the DNA. You can link it to the DNA using genetic engineering techniques of a protein you're interested in here. And here it's some protein which goes in the mitochondria. And so wherever that protein goes in the cell, when we express that protein in the cell, the fluorescent molecule goes with it, and then we use our fluorescence microscope. And so here is what you're looking at, are fluorescently labeled mitochondria in these cells, and the red is immunofluorescence, so that's staining using an anti-tubulin antibody, and so we can see the green when we take one image, and we change the lenses, we change the filters out, and we look for the red light, and then we take an image of that, and then we can see the microtubules, and we can see the mitochondria. And we've also got a blue stain here, and that's a stain called DAPI, and that's used to bind to DNA. It only binds to the nuclear DNA, so that's all we see. So here we've got a nice image of mitochondria being stained with green fluorescent protein attached to a protein which goes into the mitochondria, the microtubules in red, and then the nucleus in blue. So obviously if we treat these cells in different ways, then we can start to see how the microtubules might change, what are the microtubules doing in mitosis, how do the mitochondria change their organization in response to certain treatments, for example. We can always track the nuclei by staining the DNA. So that's a real quick overview of immunofluorescence. And next time we're just going to get into a little bit of experimental design and how we really kind of find out what we know using experimental approaches to science. And so I'll see you next time.